Welcome back to Kitchen Science Biology Lab. We're now going to go over the second law of Mendelian genetics, the law of independent assortment. In this case, I've now added a second trait. Our first trait that we looked at was the color of the fish, whether or not they were dominant trait of red or recessive trait with yellow or orange. We're now adding a second trait of whether or not they have a tail, represented by the G. So big G means they have a tail, little g means no tail. In this case, we're going to start off with our parents that again are heterozygous. So they're heterozygous for color, one red, one orange allele, and also heterozygous for tail. So tail and no tail. Same is true for mom as it is for dad. Tail, no tail, red and orange. When we go through meiosis, our law of segregation is still applying, that we need to have one copy of each trait. And so we look at our genotype up here. In mom, everybody is going to have a color trait. Everybody is also going to have a tail trait. And so our first color trait we can put out is that half of our gametes are going to have red and half of our gametes are going to have orange. Now we have to place out our tail traits. If we were to place it like this, and say that red always went with tailed and orange always went with tailless, this would be incorrect with our Mendelian genetics, the law of independent assortment. The law of independent assortment is telling us that we're going to have a lot of different combinations, that we're going to have a possibility of red with a tail as well as red with no tail. And same is true with our orange. Orange with a tail and orange without a tail. Since our dad has the same genetic makeup as our mom, I'm just going to go ahead and fill that in with our fish. I did not leave enough space on my grid to map out all of our models with fish, so I'm going to use the letters here. So now again, just like we did before, we have to copy down the columns. So in this case, this column and this column, the gamete for mom donates the capital R for the red trait. In this case, and as well as this, we have the recessive lowercase r from mom. We now also need to represent the fact that the egg contains a trait for the tail. So in this first column, we have a capital G, and we're going to leave a little space so we have room to put dad next to it. In this case, we have the lowercase g for no tail. Moving over to the next column, we have an orange gamete with also the tail. So we're going to put the capital G representing, and the lowercase g representing mom. We now need to come back into our Punnett square in our dihybrid cross and put in the information that dad is contributing. So in this case, dad is contributing a red as well as a dominant allele for tail. So we're going to have a big R, big G going all the way across this row. So big R, big G for dad. Big R, big G for dad. Big R, big G. Big R, big G for dad. This next row, we have a big R for red, but we have a little g representing our tailless condition. So big R, little g, big R, little g, big R, little g, big R, little g. Next row down, we have recessive in color, but dominant for our tailed condition. So little r, big G. Little r, big G. Little r, big G. Little, big, little, big. Our final row down here at the bottom is actually recessive for both traits. So it's recessive for our color, so little r, and is recessive for our tailed condition, so little g. So little r, little g. Little, 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 little. We now have our 16 potential combinations represented here in this grid. And so now if we look at it, we're going to have to count, and we're going to have to represent all 16 individuals. So in this case, we're going to count how many, and we're just going to look at phenotype combinations here. And when we're looking at the phenotype combinations in our grid, what we're trying to look for here is, are we going to have how many individuals are going to be red with tails. And if you are red with a tail, you need to have the red gene plus something else and the tail gene plus something else. So now we can come through and count those off in our Punnett square. So we're looking for anybody that has a capital R and a capital G. That second letter does not matter, recall, because we're looking at the dominant trait with that dominant allele. So we're just going to draw an X through everybody on here who has represented both of our traits in terms of the phenotype. So here we have big R, big G, big R, big G, big R, big G, big, 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 big R, big G, only the big R, no big G, no big R, no big R, no big R, no big R. So now we can count how many individuals in our population would be representing our red phenotype with tails. And if we count it out, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 out of 16 individuals are going to be represented by the dominant phenotypes for both traits. Now we can count how many are red with no tails. What would be the phenotype for this? If you said a capital R plus another letter and then 2 lowercase g's, you were correct. So now let's look at, we'll circle these guys, anybody who is, has a big R and 2 lowercase g's. So here we have 1, big R, 2 lowercase g's, 2, big R, lowercase g, 3. And then we have these 3 individuals remaining, none of which have a capital R. So in this case, 3 out of 16 are going to represent our phenotype of a red color with no tail. Now we can do our next phenotype of orange with tail. So in this case, orange, 2 lowercase, and then we need to have that capital G representing our tailed condition. And for these guys, let's go ahead and mark them with a triangle. Here we have our first one, 2 lowercase r's and at least 1 capital G. Here we have another one, 2 lowercase r's, capital G. And then our third, 2 lowercase r's and a capital G. So for this phenotype in this ratio, we have 3 out of 16 have an orange color with a tail. That leaves us with this last little cell here with double recessive. So it will have the recessive phenotype for color and the tail condition. And in order to have this phenotype, we have to have homozygous recessive on the color as well as homozygous recessive on the tail characteristic. And this occurs 1 out of 16 times. So you have to do a big, messy Punnett square in order to figure out what is the possible combination with our dihybrid cross. And this is something that Mendel saw with his peas and his offspring in looking at wrinkled versus round seeds, as well as yellow versus green, that he was able to determine that there are 4 possible phenotypes, and they always came out at this predicted ratio. Now, Mendel just didn't plant 16 plants to see what would happen or study 16 seeds. He planted thousands of seeds to back up his hypothesis as to exactly how it was all going to map out. And here you have the law of independent assortment with Mendelian genetics performing a dihybrid cross. Thank you.